Okay, so picking up where we left off. Okay, so last time we talked about monophyletic groups versus paraphyletic groups and also uh, polyphyletic groups. So William Henning introduced the first method for using traits to infer evolutionary relationships. And so these were based on synapomorphies, which are shared derived characteristics. So synapomorphies uh, are present in two or more taxa, and also the characteristics are found in the most recent common ancestor, but they're absent from the most distant common ancestor or from a distant common ancestor. So they allow for identification of monophyletic groups. So these are evolutionary units that include an ancestral species and all of its descendants, but no others. And so these are called clades or lineages. Some possible complications exist when you're inferring a phylogenetic tree because sometimes traits are similar due to independent evolution and not common ancestry. So when the trait is similar because of and it's due to independent evolution, that's called homoplasy. When the traits are similar and they're due to common ancestry, that's called homology. So sometimes you do get a situation where there's a reversal in a character state, in a character which means that the character appears, but then um, it's lost. And so that makes it seem like uh, it was never there to begin with. So species may be in one monophyletic group based on one trait, but a different group using another trait. So there are multiple methods used to filter probable trees. So one is parsimony. So this means that you're using the simplest explanation. So it requires the least amount of change. There's evolutionary distance, which quantifies average frequency of character changes among pairs of taxa and determines which tree best aligns with the data that you have. And then there's the maximal likelihood and Bayesian analysis. So this requires information beyond the data matrix. So Bayesian analysis uses an extra layer of probability to add another layer of probability to your statistical analysis. So sometimes when you see phylogenetic trees, they can give you different information. So if you're looking at a phylogenetic tree without a scale bar, the length of the branches are meaningless. Okay? Because there, in that case, the main point of the phylogenetic tree is just to show the relationships among the taxa. So mainly to show um, the branching pattern of the taxa. So you're just focused mainly on the tips. So the length mean nothing in that case. But if you do see a scale bar, it could be used to show the genetic difference among populations, and also it can show you evolutionary time, like these two. So in this case, the length of the branches do make a difference. Convergent evolution, this is a common cause for homoplasy. So it's an independent evolution of similar traits in distantly related organisms that's due to the organisms being adapted to similar environments. So within a species, some characters may be homologous with characteristics in other species, while others may be convergent. So looking at an example, um, if we look at the flowers and water lilies and wild roses, we could use phylogenetic evidence, uh, structural evidence, and genetic and developmental evidence to see whether the fact that they both have flowers because of homology or homoplasy. So are they homologous or convergent? So water lilies and roses are in a monophyletic group called angiosperms, where in this lineage all of the plants have flowers. So this suggests that the flowers were inherited from a common ancestor. The structural evidence is that flowers are built upon the same template, and the genetic and developmental evidence is because genetic tools and developmental patterns for flowers are similar in different plants. Sorry, are different. Yeah, sorry, I got caught up. Are similar in different plants. So here's a phylogenetic tree that shows you water lilies and wild roses. So both of these have flowers, and if you look at their common ancestor, these also had flowers. So because the common ancestor had flowers and all of the descendants have flowers, that lets you know that having flowers is homologous. So the flowers of lilies and roses are homologous. And another example, if you look at the streamlined bodies in dolphins and ichthyosaurs, so ichthyosaurs are extinct reptiles or aquatic reptiles, and dolphins, um, they look very much like dolphins. So they both have those streamlined bodies and large dorsal fin and flippers. Looking at the phylogenetic analysis, this indicates that these are not due to common ancestry because dolphins are mammals and ichthyosaurs are closely related to reptiles. And when you look at the sister groups of the animals, the sister groups lack those characteristics. So they lack the dorsal fin and flippers. So this suggests that the similarities were the result of convergent evolution. So here you see a phylogeny that shows you dolphins. So the sister groups to dolphins, primates, and rodents, they don't have the flippers and dorsal fins. If you look at the ichthyosaurs, their sister groups, lizards, they also don't have the dorsal fin and the flippers. And then if you look at the common ancestor of dolphins and ichthyosaurs, they also did not have a streamlined body, fins, and flippers. So this suggests that the similarity of the traits are homoplasy. So they're because of convergent evolution. So they just developed independently because the animals were adapted to similar environments. All right, so now let's apply what we discussed about phylogenetic trees and homologies to a case study. So here in this case study, it looks at where do whales belong in the tree of life. So hippos, cows, deers, pigs, camels they belong to a group called Artiodactyla. So that's an order. Artiodactyls have hooves and an even number of toes, and they also have an unusual pulley-shaped ankle bone that's called the astragalus. So looking at morphological data, this places whales as the outgroup to Artiodactyls because they don't have the astragalus. So they don't have ankles at all. So this would be using the morphological traits. So data set one. Okay. But looking at the second data set, so looking at DNA sequence, this data suggests that whales were a sister group to hippos within the Artiodactyl clade. And so the tree implies that there was a gain event and then a lost event. So meaning that the ancestor of whales had the astragalus, but then lost it. It was reduced. The structure, so it is even though it is less parsimonious, um, because remember parsimonious means the simplest explanation. So the fact that there's a gain of the astragalus with whales, um, and then a loss, it makes it less parsimonious. But here you're seeing the phylogenetic tree using the second data set. So this is using the DNA sequences. So DNA sequences do have the whales as a sister group to hippos. And so and then looking at a third data set that uses transposable elements, whales and hippos share several SINEs, which are short interspersed nuclear elements, and these are not found in other groups. So SINEs occasionally insert themselves into genomes of mammals. So, and the chance that the same SINEs insert themselves into the same place of a genome in two different species is very unlikely. So the SINEs are derived characters and support the hypothesis that whales and hippos are closely related. So here you're looking at a data matrix, and remember when you see a 1, that means that the character is present. If you see the 0, that means that it's absent. So if you look at whales and hippos, they have very similar uh, characters when you compare them to all other artiodactyls. Okay, so that's why they're placed as sister groups or sister taxa. So to conclude, whales are artiodactyls, and they share a recent common ancestor with hippos. So this ancestor was probably a land-dwelling artiodactyl that fed in shallow water, similar to what hippos do today. So hippos are very amphibious. They do spend a lot of time in water. In 2001, fossils of artiodactyls were found, and this supported this hypothesis because the fossils possess an unusual ear bone that's only found in whales, and also they have the pulley-shaped astragalus. So the combination of DNA sequence data and fossil data clarify the relationship between whales and artiodactyls. So we'll pause here and then we'll continue.